Okay, good morning. I'm sorry. It's me again. Okay. Next speaker is Dr. Cathy Cutler from Brookhaven International Laboratory and the chairman of SNMMI this year. Okay.
Good morning, ladies and gentlemen. I'm Dr. Liu from National Taiwan University Cancer Center. It's my great honor to chair the theranostics section of our annual meeting. With the rapid development of theranostics, we are facing multi-dimensional challenges, including not only the change of clinical practice, but also introduction of clinical trials as well as radioisotope supply. So we have invited outstanding experts from the United States, Europe, and Taiwan to share their experience in this field. First of all, we have Dr. Cathy Cutler from Brookhaven National Lab in the United States. She's also the president of Society of Nuclear Medicine and Molecular Imaging in the United States from 2024 to 2025. She earned her bachelor degree in biochemistry and a PhD in organic chemistry from the University of Cincinnati, followed by doctoral research in analytical chemistry. She has made tremendous significant advances in theranostics, developing and producing medical radioisotopes, translating them into clinical application, and then strengthening the global supply chain. She has made unbelievable advances in theranostics. Her outstanding efforts has been instrumental in advancing radiotheranostics and promoting the collaboration between research institution, industry, and clinical centers. So let's welcome Dr. Cathy Cutler from United States. Cathy, the floor is yours.
Thank you. I want to thank you for inviting me to give this talk, and thank you for the hospitality. I've never been to Taiwan before, and I've been very impressed with the food and the culture here, so I thank you for this opportunity. So today I'm going to talk to you about isotope production, but I also thought as part of the SNMMI, I wanted to talk to you a little bit about some of the things that we're doing to try to help promote theranostics. So I don't know how familiar you are with the Society of Nuclear Medicine and Molecular Imaging. Most of us know us from the annual meeting that we do every year, but we actually have a number of different things that we focus on in addition to the education that we actually provide from the meeting. And some of those I thought I would highlight here. Advocacy. We have some of the similar issues that was highlighted in some of your earlier speakers' talks, particularly reimbursement and regulations. So we have to advocate to make sure that the policies that are put in place actually help promote nuclear medicine and not go against it. So there's a lot of work we do there. We do a lot of outreach to patients as well as to public to try to educate them about nuclear medicine. And then we do quality of practice, and then we really focus a lot on research and discovery for bringing forward some of the new agents. A major focus for us right now is the workforce pipeline, and that is due to the fact the field is growing so quickly that it's been difficult to really attract the number of people that we need to really supply the workforce. And a survey that we did a few years ago has us a bit nervous because we're expecting about 30% of our workforce to retire in the next 5 years. So we're trying to work to try to make sure that we address that. And one of the other things we focus on is actually ensuring that society is meeting the needs of the members. So we constantly look at what our members are needing and if we're actually meeting that and changing the society to help do that as we move forward. So one of the big issues that we face is most people don't know what nuclear medicine is, and often they're a bit terrified by it. So what we do is actually try to work on communicating to the public and developing communications across all audience to really kind of educate them about the value that nuclear medicine actually brings to their lives. One of the ways that we do that is actually focus on PR. So we focus on major stories that we can actually put forward each month. So for instance, in the US at least, we have like Breast Cancer Month, Prostate Cancer Month, Neurology Month. And so we help focus on the things that nuclear medicine can bring forward in those selective areas so that we can highlight to the public what we're actually bringing forward. And some of them are other stories, like we had a story in which we showed that patients who used hearing aids, it greatly reduced dementia. And you could only see that through using nuclear medicine imaging. So this really kind of helped highlight to people what the value is, and it's actually been very successful, and we're trying to do a number of things in multimedia to try to really educate and bring forward the value. The other thing we do is a lot of advocacy. And as was highlighted in the talk by the NHI that was given earlier, we really need to work with our government and our agencies to make sure that they understand what they're implementing and the impacts on that. And so one way we do that is, for instance, every year we actually have what we call a Capitol Hill Day. And so we actually go to Washington, D.C., and we bring in both physicians, patients, people from industry, and members of the society. And we go in and meet with our legislators to talk to them. And we go in, we usually have 3 issues that we focus on. Last year, our issues were reimbursement, research funding, and there were some issues coming forward that would be against nuclear medicine, and we were not in favor of those. One of the things we did that was new last year is we want to try to create champions in the legislature that will help promote nuclear medicine. And so Representative Neil Dunn was one who helped us with reimbursement efforts. He put forward bills to make sure that radiopharmaceuticals were reimbursed at the correct level. And so we awarded him for his efforts. He was actually a nuclear medicine physician that had been part of the society, and so it was nice to give him that award. The other representative that we awarded was Din Barry, and he was actually helping us put language in, nuclear medicine language, to ensure that theranostics was something that would get funded under research efforts. And so this language was added to the Department of Defense and to the National Institutes of Health. We also have a great outreach to patients, and so we have a patient outreach, a patient advocacy advisory board, where we meet with a number of patient groups, and then we have materials on the website to help train them and give them advice about what they should expect as they go through their procedures. At the SNMMI, we have an annual patient education day, so we invite patients to actually come in, and we have sessions focus on them. They get to go see the exhibit hall, and then we actually meet with them as leadership so we can understand their needs and how we can help them and they can help us actually as we move forward. Some of the other things we do is actually reach out to other communities and societies to educate them about the value that nuclear medicine can bring to their practices. So we work to set up sessions at other meetings. We do live education roadshows and work with them to develop fact sheets so that they can understand what goes forward. And then one of the things we did 2 years ago is we had a clinical trials network that focused on diagnostics. As you know, we're going into theranostics and therapy, so we did set up the Therapy Clinical Trials Network. And what we're seeing is that there's a lot of companies that are getting involved in therapy, but they're very new to nuclear medicine. So they don't always understand how best to set up their clinical trials, what they need to consider for calibrating their equipment, and what they need to do to consider photosimetry. So this helps companies that are interested to find centers that are set up to identify those that have the services that they need so they can basically move forward with their clinical trials. One of the new things that was put in place was the precision dosimetry imaging biomarker. It's called PBIB. This was actually set up through the FNIH. It's funded by industry, and it's being organically administered by the Society of Nuclear Medicine and Molecular Imaging. And the goal of this is to establish secondary standard laboratories. As we go into therapy, it's very important that we know exactly what the activity is that we're injecting. And we found that there was a lot of variability. So we're working to try to narrow that variability and make sure patients are aware, our physicians are aware exactly what they're injecting. We're trying to harmonize SPECT CT sensitivity and then standardize standardize the dosimetry calculation workflows. And this is an international effort where we're working with other societies to help put this in place, because we think this is very important to ensure the success of therapy as we go into more and more clinical trials. So right now, I think is a very pivotal time where we're seeing a lot of advances in the science and practice, and theranostics is really gaining momentum. There's a lot of new research that's coming forward that can have a high impact on patients, and we're working to try to ensure that that happens. One of the things that we did last year in the May is we had a theranostic stakeholder summit. And this is because largely nuclear medicine has been focused on diagnostics, as it was pointed out earlier when you opened the session. We're now changing in which we're doing more therapy, and this really changes how we implement our practice. And so we need to focus on what do we need to change to ensure the success as we go forward. So we had a summit in which we invited industry, we invited other societies to come in, and this was the reason that we did this is that we wanted to identify what the major challenges were, the barriers that we needed to address, and we needed to see the whole picture. And if it was just us talking to ourselves, it would be a very narrow picture. So understanding what industry was seeing, what other societies were seeing, helped us to identify some of the things we needed to work for. As you heard earlier, reimbursement. We need to ensure we have appropriate reimbursement for both the radiopharmaceutical as well as for the physicians and our practitioners who are implementing this. We need to work to ensure that we have the workforce so we can actually have the quality people in place to help do this. Infrastructure. As this grows, we need to make sure that we have the appropriate infrastructure. This includes the isotopes that we need to actually implement this. And we need to make sure that people understand the value. So we need to build bridges with other practitioners so that they will promote nuclear medicine and bring us forward into the field. So some of the ongoing challenges that came out of this is workforce shortages. We see gaps in training and education. People aren't being trained on theranostics, and they're going into practice not necessarily knowing what that is. Part of what I'll talk about today is some of the isotope supply issues that we have. Without the isotope, you don't have your radiopharmaceutical, and we can't train. So this is something that we need to address. In the U.S., we're facing, because of our current administration, some research funding limitations. As we're going forward and developing these more complex radiopharmaceuticals, it's important that we have the research to develop this, but also to support the clinical trials as we go in to make sure that these move forward. And then we need to ensure patient access. And this is particularly challenging in some of our underserved rural areas in the U.S. So how do we move the field forward to make sure that these patients have access? So on the isotope supply chain, isotope supply has basically been a global collaboration that we've really worked together to ensure that we have the isotopes that we need. That being said, this often results in that we don't have the isotopes we need domestically. And this is for a number of reasons, and this is something that we need to work on as we move forward. And part of what we need to do is ensure that policies are put in place that will actually support domestic, resilient, robust supply so that when we need the isotopes, they're there in the quantities that we need to move it forward. So how are isotopes produced? And so isotopes are produced using nuclear reactors, using neutrons to irradiate targets. We have accelerators where we use beams of protons, deuterons, and various things to bombard targets. Or we can utilize generators. And generators allow us to provide isotopes in which we have a long-lived parent that decays to a daughter, and we can ship these around to remote areas, and you don't necessarily have to have a reactor or an accelerator on site. Now, the concept of the generator actually was developed at Brookhaven National Laboratory, and it was developed by Marjorie Green and Walt Tucker. And they were actually working on an iodine generator when they came up with this. And there was this pesky impurity that they couldn't figure out what it was that was messing up their results. And it turned out it was Moly-99. And they realized that Moly-99 could decay to technetium, and that technetium had ideal properties for doing nuclear medicine. And so Pal Richards, who was the chair at that time, spent about 20 years really promoting the use of technetium and went out to societies indicating this good use. At that time, they were operated by what was called the Atomic Energy Commission. They put forward a patent for this generator, and basically the A said there was never going to be any commercial use for this, so they would not award a patent. That being said, these generators are now used in about 20 million diagnostic investigations around the world. They then went on to actually look at how do we take this technetium that we get off this generator, which has a very rich chemistry, and develop drugs that we give them to properties that they will localize where we want them to. And what originally happened is they had scientists like me who would come up with their particular recipe to develop these drugs. And the challenge was it wasn't uniform. So Bill Elkelman worked with Pal Richards to come up with what they called the kit. And so the kit was a vial that contained the ingredients to actually formulate that drug. And this allowed you to make sure that every site was formulating the same drug so you could move forward and actually go into clinical trials. The other thing that it did was actually provide a product that companies could then sell, which actually helped promote the economic interest in this field. They then went on to develop FDG. There was a physician that was working at the NIH, and he was interested in imaging metabolic disorders in the brain. And he realized that glucose uptake varied, and that if they could label glucose, this could be used as a tracer. So he approached Al Wolf at Brookhaven National Laboratory and asked, would you be willing to work on this? And he said, I will, as long as we can translate the knowledge to everyone, which I thought was actually very important. Here you can see Al Wolf, who was working with Tatsuo Ito and Cian Wan, who basically developed the chemistry, along with Joanna Fowler, to incorporate fluorine-18 into FDG so that it actually could be used for imaging. And now we see that there's roughly about 1,500 PET scanners around the world that are predominantly used for actually imaging FDG. After this, there was the realization from Prantika Sam. She was a veterinarian that worked at Brookhaven, who worked with Dr. Hal Atkins and realized that FDG was looking at metabolic disease, and that you could basically use this to evaluate different things. So they actually did a study in which they used FDG to look at different cancers and showed that you could use it for both diagnostics, but also to monitor response. And this is what FDG is most commonly used for today. And here you can basically see when FDG was actually developed in 1980, and it kind of moved along, but when it really took off was when we got proper reimbursement around 2000. And so it's really important that we get proper reimbursement to actually help nuclear medicine grow. So where are we today? So today we're moving from these small molecules in which we're doing theranostics, and which we're taking targeting molecules and we're attaching isotopes, first for diagnostics, so we can determine if the disease is there and if the target is there in high enough quantities to treat, and if the normal tissue uptake is low enough so we can move forward. And if so, then we can go on and attach a therapeutic where we can then go on to actually treat these patients. And Dr. Baum has been one of the leading people in this field, and I look forward to his talk next. So based on more or less the growth of theranostics, it's projected that we're going to see significant growth over the next 10 years. And we're also seeing that there's a number of drugs that are actually entering into clinical trials. So here's a slide that basically shows a number of different clinical trials are ongoing, showing by preclinical, phase 1, phase 2, and phase 3. And so you can see that the amount involved is actually very high, and companies are spending billions of dollars to actually get these drugs moving forward. Now, here's a list of isotopes that are currently being investigated in clinical trials. And here you can see that on the diagnostic side, gallium-68 right now is the highest used diagnostic, followed by zirconium-89 and fluorine-18. And we see that things like copper are actually coming online, and lead-203. And this is important to understand the diagnostic side, because at least in the US, normally we start with the diagnostic and clinical trial because the FDA requires it, and then we move on into the therapeutic. If we look at the therapeutic side, we see that lutetium-177 has the largest number of clinical trials, and that's based on the success of what we've seen from the approved agents. But we see that the alpha emitters are quickly becoming behind with actinium-225, lead-212, and we also see copper-67 now with availability that's really coming up. So we're seeing a large growth in this field, and we're seeing that the number of isotopes entering this is growing as we move forward. Now, this is a slide that's provided by Oppenheimer that kind of looks at the different agents that are in the different stages of preclinical, phase 1, phase 2, phase 3, and approved. And so you see there's a huge number of agents that are coming forward. And the question that comes forward is, are we going to have the isotopes to supply these? And so hopefully I'll answer a little bit of that as we go through this talk. So some challenges that we actually have with our isotope supply. In the US, we actually import most of our isotopes from outside of the country. And we actually have 30 medical isotopes in which we are completely dependent on external supply. So the other challenges we have is that reactor outages, and these reactor outages can result in shortages, particularly of technetium-99m, iodine-131, and lutetium-177. We have reimbursement complications. So we're not getting reimbursed on the correct level for the radiopharmaceutical, for our physicians, as well as the isotope itself. There are regulatory issues as we move into this. So therapy is new, and agencies are grappling with how to handle this and how should they regulate it. We're seeing in the US that the FDA is working with us, but they're having challenges on understanding how we move forward with these therapy clinical trials. The NRC is looking at the regulations, and we're having to work with them to make sure that they're not implementing things that could actually inhibit patients having access to this. Transportation, as we get into therapy, we're shipping higher quantities, and one of the things we recently saw is that the IAEA changed the values that you use for shipping. And this could have a huge impact because it means that you will have to transfer from using a very simple commercially available container to a highly complex type B container in which it costs a lot. You have to have a quality assurance and maintenance program in place, and very few people actually have that, and we actually don't have enough of them to support this. So we have to work to kind of minimize that. And then work for attracting the brightest into the field to make sure that we have them as we move forward. And additionally, as we have new people who are entering the field who aren't familiar with it, we really need to educate them to understand the quality issues and the things they need to do for supplying this. So there was a paper that recently came out from Theranostics that kind of highlights some of the challenges that we actually have with production. And the paper highlighted isotopes, how those isotopes are made from reactors, accelerators, the energy of the accelerators, how they're basically used, and then kind of highlighted what the number of reactors are. So total reactors they see is about 222. Out of that, there's about 30 that can actually be used for isotope production. And so one of the things you see is that actually in Asia, this green color means you guys have the highest number of reactors. So you're actually doing pretty well. Actually, though, those of us in North America who are one of the biggest users, we have the lowest number of reactors, and this is why we actually end up importing many of those radioisotopes. But this is important as we see that there are a number of continents that really don't have the reactors that they actually need to supply this. Now here you can see cyclotrons. Again, this is from that same paper. And again, you see that in Asia, you're actually doing really well. You have the highest number of actually cyclotrons that are available. And I think because you have these resources, this is one of the reasons why your area is predicted to have the highest growth in the next 10 years in this field. Now, North America, we're doing a little bit better with cyclotrons. You can see that we're getting closer to that green. But one of the other things I want you to look at is the number out there. So to really support nuclear medicine, we have about 1,500 cyclotrons currently in the world. So it takes many more cyclotrons to help do the production than reactors. And that's an important thing to consider. Now, there are new technologies that are coming online, new accelerators. There are electron beam accelerators. We have the IBA Rototron, and this allows you to do photoelectron reactions, and these reactions can produce isotopes in higher yields and purity than we can get from other types of accelerators. And it's very exciting that these are coming online to supply some of the isotopes that we're having shortages with. We also have new LINACs that are coming online. NIOWAVE has built a LINAC, and Nusano has a new one that are coming. And these are helping produce higher amounts of isotopes as we're coming into this. So here are some of the therapeutic beta emitters. Beta emitters have been those that have been most prominently used, basically because they have been readily gotten at. Lutetium, as we've talked about, has been the most heavily used. Copper-67 is one that there's been interested in for a long time, but it's only recently that the production has gotten to the point that it actually can support clinical trials. Scandium-47 is one that's similar to lutetium. It's got some enhanced capabilities such as the imaging isotope and others, and the fact that it has scandium-44, which you can use for imaging. So there's been a lot of work in looking at producing this. And then terbium-161, which has similar chemical properties to lutetium, but it has nuclear properties such as increased Auger and conversion electrons that have been demonstrated to give a higher effective dose compared to lutetium. So when we look at lutetium, lutetium was first kind of made available at the University of Missouri, and that's where I worked to kind of bring this online. And lutetium, the direct route is very easy to make. You just take enriched lutetium-176, you put it in the reactor, it absorbs a neutron, you get lutetium-177, and you can get about 20 curies per milligram because it's got a very high cross-section. And then you just dissolve it up and you can ship it out, and it's a very easy reaction. The challenge is that you end up making the long-lived lutetium-177M. And this can be a challenge for people to handle. So they started looking at alternative ways to make this. So the other way you can make it is the indirect route, irradiating a terbium-167, a terbium-176, which forms a terbium-177, which in 1.9 hours decays to lutetium-177. Then by separating the lutetium from this target, you have a high specific activity lutetium without this long-lived impurity. And so this is what people are moving towards and what mostly is used right now. The challenge is that the cross-section from this drops from over 2,000 down to 2.8. So what this means is that I've got to irradiate 1,000 times more target material to get similar production yields. And I have to do a very complicated separation of that lutetium from the ytterbium. So this production route requires much more production capability, and it requires higher input because of what it takes to actually make this. That being said, there are a number of reactors that are now producing this, and it is actually out there. And this indirect route is actually what's being evaluated for other production, mostly because it gets rid of these long-lived impurities and gives you a high specific activity material. So lutetium has gotten to the point that there are now a number of different companies, and I'm sure there's more than what I've listed here, who are actually supplying it. When it first came out, it was approved in Lutathera, and it was as the carrier-added material, but they're moving towards the non-carrier-added. The Pluvicto was approved with the non-carrier-added, and most of the things moving forward will be using the non-carrier-added lutetium. Now, one of the things I think is exciting is that people are a bit concerned about the number of clinical trials, but people are looking at more diverse ways of bringing this online. And so I think one of the unique things I've seen is that Bruce Power in Canada has come up with a device that allows them to use power reactors to actually produce lutetium-177. So this means they don't have to build a new reactor. They can utilize existing reactors. They don't have to pay for the infrastructure, but they now have increased the capacity, and they've been making lutetium-177 in this way, as well as Moly-99, and they're working to expand this. And I think this is actually a very exciting way that may help us actually meet the demand. Now, copper-67 is one that's been of interest for some time. The DiNardos out of California were looking at labeling antibodies and went into clinical trials. And they were being supported by national laboratories that were producing the copper-67 on proton accelerators, but they could not get enough, and they actually had to shut down their trials. So it really changed it for copper-67 were these electron beam accelerators that could actually produce it. You get it in a higher specific activity and yields. The Idaho Accelerator Center was really the first to bring this online, but now there's a number of companies that have joined this, and so we're seeing this actually getting into clinical trials and being expanded. The nice thing about copper-67 is you have the copper-64, which you can actually use as the diagnostic. Now, another one I talked about is scandium-47. Scandium-47 has a number of different routes. You can actually have a generator route that produces this. You can produce it with proton accelerators, but probably one of the most exciting reactions is actually the electron-photon route, which can produce it in very high specific activity and clean. This has been developed out of the Argonne National Laboratory and should be brought out onto the market soon. So we will see how this works as it moves forward. So the nice thing about scandium-47 is you have scandium-44, which has about a 4-hour half-life. Scandium-44 can be produced on cyclotrons, or there's actually a generator that's available. And here you can see some work that was done at PSI in which they compared the imaging. A is the imaging from scandium-44, B is the imaging from scandium-47, and you can actually see that they're very similar. So this is very nice as you actually get started and go into clinical trials. And then finally, terbium-161. So terbium-161 is of interest, as I stated earlier, because it has additional Auger and conversion electrons. And there's been work that has demonstrated that you can get higher efficacy. Now, the way that you make terbium-161 is similar to the indirect route used for lutetium, in which you take enriched gadolinium-160, put it in a reactor, you form the gadolinium-161, which then decays to terbium. You then do a separation, and then you get your terbium-161. You can also make this using deuterons, and there are people looking into this route, and thus far I've only seen it feasibility studies, so I'm not sure how well this will work. But there are a number of reactors that are actually bringing this up. It actually is in clinical trials in Europe, and availability is starting to be available actually in the US. And here's some work that was done. This is a somatostatin analog, which they attached the terbium-161 and then went on to actually do therapy studies. And you can actually see the uptake with the gallium, and then you can actually image the terbium-161. And basically, the studies thus far have shown the higher efficacy of the terbium-161. So then alpha emitters, there's a number of alpha emitters that people are interested in and looking at. Part of the challenge is that the production of this has mostly been from harvesting from old materials. And those materials are no longer available, so we've had to come up with other ways to actually produce these alpha emitters. So actinium-225 is one that is of great interest, and it's originally produced from the decay product of long-lived thorium-229. But now there's a number of different reactions that people are evaluating for actually producing the actinium-225. And here you can see in the different reaction pathways, I've listed the companies and labs that are actually looking at producing this. So I would say they're slowly bringing up these lines, but I think that within the next 2 years, we're going to see a dramatic difference in the amount of actinium-225 that's actually available for supporting these clinical trials. Astatine-211 is another alpha emitter that's been around for quite some time. It's produced by using a cyclotron with an alpha beam by irradiating bismuth-209, and it requires at least a 26 MeV energy alpha particle beam. You can also look at production using a generator using radon-211. This was originally suggested by the group from Triumph, but there are others who are utilizing new MOFs that are looking at supplying this as generators. So some of the challenge is that we don't have enough of these machines, but there are networks that are being developed, and there's the World Astatine community that is really working to try to share this technology and develop these regional sites to promote Astatine-211. And there are commercial companies that are coming online, at least in the U.S., the Nosano. This is their lead product that they're looking at, as well as Ionetics that has a 30 MeV cyclotron, and they're expecting to have commercial production within the next year. And then finally, lead-212. So lead-212 basically comes from the thorium-228 feedstock. Thorium-228 was originally derived from environmental waste materials, which is, there's very little of it. But it turns out you get a large amount of thorium from irradiating radium-226. And so you can get this material and then utilize it for making lead-212. And it has one of the highest cross sections of this, so you actually make a lot of it. So this is really not limited in supply, and there's a number of companies that are now coming online that are actually supplying the lead-212, and I think this is very exciting as we move forward. So in conclusion, there's a lot of growing interest. We have a lot of companies who are spending money, a lot of people who are actually looking at developing this. And although right now we do see there's some limitations in the supply, I think that the supply chain is starting to diversify and it's becoming much more economical. So we're seeing companies actually enter this space and make these. And I think that is really going to change the availability that we have going forward, and so that we will start seeing a more robust supply of these isotopes that will help supply so that we can actually move forward in this area. And I also think a lot of national labs are actually putting forward to try to help support these companies as well as to help support their countries to ensure that they have this. And in that, I thank you.
Thank you. So because of the time, we are moving to the next speaker. Okay, if we still have a little bit of time, if there, okay, come, please come.
Please take a photo with our moderators.